In this lecture, we'll go through blood supply to the brain, brainstem, and a bit of the spinal cord as well. We'll start with arterial input, then our low resistance drainage and how we regulate input in an activity-dependent manner. And finally, we'll go through the finer features of the blood-brain barrier. In the end, we'll try to wrap it all up with some simple schematics of the circle of Willis and the vascular territories of the major blood vessels. So the brain is an expensive organ. Neurons are always active. Whether you're thinking hard on something or just sitting there daydreaming, your brain is always working. If brain doesn't work, you don't continue to live. At least you're not aware if you're alive. So the blood is going to pretty quickly move from the heart to the brain. And the brain is supplied by a few different arteries, which will then branch and converge to form three major cerebral arteries, and then a few different arteries to serve the hindbrain. So from the aorta, the external carotid, the common carotid is going to branch and form the external carotid, which we don't care about, and the internal carotid, which does matter to us. All right, so here we've got our aorta, common carotid here, branching to serve the outside of the head there. We don't care about that external carotid. We're going to follow the internal carotid on up and serve the brain there. We'll draw this out in just a little bit, but the internal carotid arteries are going to have a couple of branch points before they finally hit the circle of Willis. There's the ophthalmic artery to serve the eye and nearby structures. There's an anterior choroidal branch that'll help create that choroid plexus that makes all the CSF for the brain. And then it eventually runs into the circle of Willis. Now, the other way that we get blood coming in is through the vertebral arteries. The vertebral artery here is a branch of the subclavian artery, so we have to take just one step further in the aorta. The subclavian then branches, forming the vertebral, which is going to sneak up and create the blood vessels for our hindbrain, as well as, of course, the spine. The vertebral arteries come together and form the single basilar artery that then runs into the circle of Willis. So here we have our vertebral arteries coming on up, converging to form the basilar artery here. This will eventually merge with offshoots of the internal carotid to form the circle of Willis, which we will draw out in a bit. Here's a simplified schematic that we're going to try to recreate on our own later. So the circle of Willis is a really nice structure here. So imagine that we have an occlusion anywhere within that circle. Well, we're not going to lose blood supply. Let's say we have a clot here in this posterior communicating artery. Well, we still have ways for blood to get around, right? This internal carotid could provide blood to the posterior cerebral artery here. Of course, this has a backup from the basilar artery, but we can head around the whole circle and nothing is lost. So this creates essentially a little insurance policy to ensure that we have adequate blood supply to the brain. Now, the internal carotid artery, aside from having all those other branches that we don't care that much about, the ophthalmic and anterior choroidal artery, the internal carotid is going to branch to form the anterior cerebral artery and the middle cerebral artery. And for most people, the internal carotid continues along as the middle cerebral artery. This is why the middle cerebral artery is a more common site for stroke, because any blood clot floating in the internal carotid is going to continue along the middle cerebral artery. It branches then to form the anterior cerebral artery, as well as the posterior communicating artery. Now, the posterior cerebral artery, the third of our cerebral arteries there, is usually a branch of the basilar artery here. Now, there are tons of other branches which we will get to in a bit. To finish off the circle of Willis, we have to connect our two anterior communicating arteries. Anterior cerebral arteries. So here's one, here's the other, and they're connected by the anterior communicating artery, of which there is only one. Then we have the posterior communicating arteries, 1 and 2, connecting our middle cerebral artery to the posterior cerebral artery. And now we have our good old circle of Willis. Now those three cerebral arteries serve slightly different areas of the brain. Here we can see their superficial areas served. So the middle cerebral artery is going to serve all of our lobes, but the most lateral aspect of them. Notice if we look at the medial surface of the brain down here, we don't see very much of the middle cerebral artery. Now, then we have the anterior and posterior. Their name tells you where they serve. The anterior cerebral artery serves the anterior portion, and the posterior cerebral artery serves the posterior portion of the cerebrum. So that means that the anterior cerebral artery is going to hit more of the frontal and parietal lobes, hitting a large part of the corpus callosum here. And the posterior cerebral artery is going to hit more of the occipital, a bit of parietal, and temporal lobes here. Notice it also serves the thalamus. Now, each of these cerebral arteries has a deep branch. Here, we're just looking at the superficial branches. The deep branches serve more internal structures, and that would be the thalamus, shown here, the internal capsule, and the basal ganglia. So the anterior cerebral artery, superficial division shown here in this cross-section. The deep division is called the medial striate arteries. The medial striate arteries serve the internal capsule as well as portions of the basal ganglia, controlling face and arm movements. The middle cerebral artery branches to form the lateral striate arteries. Notice, slightly more lateral than the deep division of the anterior cerebral artery. The lateral striate arteries serve then similar areas, internal capsule, basal ganglia, but this time dealing more with legs. The posterior cerebral artery's deep division serves more of the thalamus and a bit of the choroid plexus. Those are called the thalamo-perforating arteries. So each of our three cerebral arteries has a superficial and a deep division. The superficial division is just called the anterior, middle, or posterior cerebral artery. The deep division has their own unique name. Blood supply for the hindbrain and spinal cord arise from the basilar and vertebral arteries. The basilar artery, shown here, is a single artery that then branches off to serve the pons. Of course, it branches to form the posterior cerebral artery. We've already covered that. Then we have pontine arteries to serve the pons here. Before that is the superior cerebellar. And below the pontine arteries, we have our anterior, inferior cerebellar arteries. In most folks, the posterior, The posterior inferior cerebellar arteries arise from the vertebral arteries, not the basilar artery. So after we've branched off our anterior inferior cerebellar artery, now we are branching into the vertebral arteries. The vertebral arteries form the anterior spinal artery, the posterior inferior cerebellar artery, and the posterior spinal arteries, at least in some people. In the majority of people, the posterior spinal arteries are a division of the pica, the posterior inferior cerebellar artery. The spinal arteries are either the single anterior spinal artery, which we can see is going to serve the front and sides of the spinal cord. Notice it's a single artery here. And then we have the paired posterior spinal arteries, which will each serve about a half of the posterior columns and dorsal horn. This gives us our typical anterior cord syndrome if we have a stroke in the anterior spinal artery versus posterior cord syndrome for the posterior spinal arteries. So that's the input. We need constant blood supply so we can pay the bill, but we don't want to have any buildup in pressure. This means we must have low resistance drainage. Two ways of accomplishing that. We're going to modify the veins, and then we're going to create large sinuses that we can dump blood into. This way we can pump as much blood as we want to into the brain, but we'll never risk building up pressure. Now in the periphery, veins have valves to create unidirectional flow. So blood can flow up, for example, but not down. When it flows down, it closes the valve, and this will allow blood to move against gravity. This is great, but it creates a possibility to have resistance. So we don't see valves in the veins of our brain or spinal cord. This way, there's no chance of having any blockage for blood flow. It's free to flow. So this lowers resistance, but what it also does is make it easier for cancer cells to metastasize. So they can freely move through, for example, spinal veins, moving from the lungs, then into the spinal cord. So this is a common cause of cancer within the nervous system, these valveless veins. The veins are connected to these large drainage sites, the sinuses, and those are shown over here. We have a few sinuses that we'll walk through. So here's the superior sagittal sinus, called sagittal because it's at the midline. Inferior sagittal sinus, a straight sinus connecting them at the confluence of sinuses. All veins drain into sinuses, and those sinuses are where we separate the two layers of the dura. So remember, there's a periosteal layer that stays on the bone, and there's a meningeal layer that stays along the brain. When the brain is not tightly opposed to the skull, this is where we have our sinuses. Now, just like our veins, we have no valves. We also lack any musculature here, so this is just a big, wide open site that we can dump blood into. So we bring blood in the arteries, and we'll drain them along the veins. There are, of course, superficial and deep veins, because we have both superficial and deep arteries. And the names make sense. Superior veins are going to drain the top of the brain. The superficial middle veins drain the middle of the brain, and then the inferior veins drain the lowest parts of the brain. And all of these veins drain into their nearest sinus. For example, the superior veins here drain into the superior sagittal sinus. The middle vein is connected to the superior and transverse sinuses through the anastomotic veins, and of which we have a couple here: a superior anastomotic vein, an inferior anastomotic vein, and that allows this middle vein here to hook up with the low resistance drainage of either the superior sagittal sinus or the transverse sinus. Now we also have deep veins to serve the internal structures, and these simply drain into the straight sinus. That's shown in portion B there. Here we can see a slightly better view of this. So those deep arteries are drained by deep veins. So here all of the blood supply to the thalamus, basal ganglia, then drain into the internal cerebral vein, and these come together to form the great cerebral vein. This will then drain into the straight sinus. All right, let's go through our sinuses. So the superior sagittal sinus is created by the separation of the dura on top of the brain. So the two cerebral hemispheres, tucked between them, we find the superior sagittal sinus. The inferior sagittal sinus is found below there. That's this kind of lighter sinus shown here. And those meet up with that great cerebral vein that we just mentioned for the deep drainage. So inferior sagittal sinus, the great cerebral vein merge together to form the straight sinus, and that allows this blood drainage to come together with the superior sagittal sinus at the confluence of sinuses. Now at the confluence of sinuses, we see a number of sinuses coming together. The occipital sinus here, the superior sagittal sinus, the straight sinus, and then the transverse sinuses, which run around the top of the cerebellum. The occipital sinus runs between the two hemispheres of the cerebellum. Those transverse sinuses then meet up with the sigmoid sinuses, and finally we hit the internal jugular, and we can drain the blood out of the brain. Now the internal jugular is also receiving input from the inferior petrosal sinus here, and indirectly from the superior petrosal sinus, which meets up with the sigmoid sinus here. And these two sinuses, the superior and inferior petrosal sinuses, drain the cavernous sinus. Now the cavernous sinus has a number of other structures that we should be aware of. So first of all, the cavernous sinus surrounds the pituitary gland. So this is where we dump hormones from the pituitary gland, is into the cavernous sinus, which then drains into the jugular vein, directly through that inferior petrosal sinus or indirectly through that superior petrosal sinus to the sigmoid sinus. So this is a site where we have hormone release. We also have a number of cranial nerves running through the cavernous sinus. Cranial nerves 3, 4, and 6 to control the eyes, as well as 2 divisions of cranial nerve 5, the trigeminal. So the ophthalmic and maxillary divisions are going to serve the top and roughly middle of the face there. We also have the internal carotid artery running through the cavernous sinus. So if we have any kind of hemorrhage of the internal carotid, we are then going to damage cranial nerves 3, 4, 5, and 6, giving us problems controlling our eyes and feeling things in the face. Stimulation of cranial nerve 5 will also give us headache, and that's what we see here. This lady had a fall that then led to a carotid aneurysm within the cavernous sinus. As a result, she had trouble opening her right eye, as you can see here. She also has a blown pupil, as we can see in panel B. Along with this, she had reduced sensation in her upper face, as well as headache. So certainly we have low resistance drainage. That way we don't build up any pressure. We can also prevent a buildup of pressure by properly regulating blood flow. Now, at a global level, we regulate blood flow through the baroreflex. We should recall this from previous lectures. If there is an increase in blood pressure, baroreceptors in cranial nerves 9 and 10 sense this, stimulate the baroreceptor nucleus of the solitary tract nucleus, triggering parasympathetic output from the dorsal motor nucleus of the vagus to slow heart rate and thus decrease blood pressure. If there's a drop in blood pressure, the opposite occurs. We don't stimulate parasympathetic output. Instead, we're going to disinhibit sympathetic output to increase heart rate. So the baroreceptor just globally changes how much blood is moving around and thus how much blood we're delivering to the brain. We also sense pH, and if carbon dioxide levels build up, pH drops. We should, of course, remember this. We'll form carbonic acid with carbon dioxide and water. Well, hell, I guess I can do that. So carbon dioxide plus water will produce carbonic acid, which quickly dissociates to form bicarbonate and a free proton. And that right there explains why an increase in carbon dioxide drops pH. Now, the pH level relates to the amount of calcium present in vascular smooth muscle cells. And that's what we're looking at over here. So what we should appreciate here is, well, first of all, let's get all this junk out of the way. What we should appreciate is on the y-axis here. This is just looking at pH. And what they're showing us is that if they dump some ammonium chloride on, it lowers the pH. Okay. I'm sorry, it raises the pH. That's just allowing us to then interpret the real figure that matters, the one on the bottom. So we're going over time. At about 2 minutes, they dump on some ammonium chloride, which raises the pH. And notice that increases the amount of intracellular calcium. That's what's shown on the y-axis. Intracellular calcium causes constriction. So this is the opposite of what happens when carbon dioxide builds up, right? If carbon dioxide builds up, then there should be a drop in pH. If we drop pH, that decreases the amount of calcium that's floating around, causing, instead of constriction, dilation. I've chosen a very poor spot to write that. And that dilation is triggered by that drop in intracellular calcium. So if our blood gets acidic, because we're building up carbon dioxide, let's increase blood flow so we can move blood around, get more of it to the lungs and exchange gas, and bring more fresh blood to the brain. These are global levels. Now, the brain also regulates where blood goes to it on a smaller level, which neighborhoods of the brain are more active, right? If it's a visually demanding task, occipital lobes need more blood. So we can have local changes in blood flow based on how active neurons are. So first of all, the good old pH carbon dioxide thing, that still works, right? If we have an area of the brain that's more active, it's making more carbon dioxide, and its local pH will drop, causing the smooth muscle and pericytes in the area to relax, increasing blood flow. So this aspect is the same. Nothing new here, just on a smaller scale. In addition to creating carbon dioxide, active neurons also spit out potassium, right? Remember, every time we fire an action potential, we spit out a bunch of potassium. If we increase extracellular potassium, we might expect depolarization. And that is true for neurons, but not smooth muscle. Notice their resting membrane potential, far depolarized compared to neurons. So they don't have quite as much of a potassium leak. So if we increase extracellular potassium over here, bumping it up to like 15 millimolar, okay, sure. We're going to depolarize the reversal potential of potassium. Right, so now the potassium reversal is going to be somewhere in the ballpark of negative 60 millivolts. That says millivolts, I promise. But what we've also done is dramatically increase our permeability to potassium, which we had very little of at rest. So sure, our reversal potential has depolarized a bit, but it's still hyperpolarized relative to our rest. So that increase in permeability causes us to move toward the reversal of potassium, which is still hyperpolarizing. So, surprisingly enough, when you dump a bunch of potassium onto a smooth muscle cell, it hyperpolarizes. And that's because they have potassium-gated potassium channels. You heard that right. These inward rectifying potassium channels here are opened by extracellular potassium. So that increase in extracellular potassium stimulates potassium channels to open up, increasing potassium leak of the smooth muscle. So if we have a look at the data up here, ES means electrical stim. So they stimulate a little bit of brain tissue there, and they have a look at the diameter of blood vessels. Control, nothing has been done here. Do some electrical stim, and notice we vasodilate. Why? Because we increased extracellular potassium and we hyperpolarized the smooth muscle, causing it to relax, increasing blood flow to the area. We know this has to do with potassium because if we apply barium or tetraethylammonium, we don't see this same vasodilation. Yeah, it kind of jumps around a bit, but it stays pretty close to 3 to 4 micron diameter. It doesn't vasodilate to 5 to 6 microns. Okay, so that's kind of weird if you only think about the Nernst equation. But if you remember the Goldman-Hodgkin-Katz equation, the buildup in extracellular potassium moves us toward potassium's reversal in smooth muscles. So they have this kind of surprising hyperpolarization. Okay, so active neurons spit out carbon dioxide and potassium, and it turns out they also spit out nitric oxide. So when a neuron is active, that calcium stimulates nitric oxide synthase, which creates nitric oxide from arginine, and nitric oxide freely floats to nearby smooth muscle cells. Now, sure, in the cartoon, these are endothelial cells, but you could call this a neuron. You just have to change the name from nitric oxide synthase to neuronal nitric oxide synthase. Let's do that now. This is typically done with a little tiny n there, and there you go. Now it's a neuron. Everyone's happy. So what nitric oxide does is stimulate this soluble guanylyl cyclase. It's kind of like adenylyl cyclase, but it doesn't make cyclic AMP. It makes cyclic GMP. Okay, then what the hell happens? Well, we turn on protein kinase G. This is the central star here. So this kinase does a few things. First of all, it causes a little bit of relaxation by increasing potassium permeability. So again, we move toward potassium's reversal. In other words, we hyperpolarize. We also decrease the amount of calcium floating around in smooth muscle by inhibiting calcium channels, stimulating calcium pumps, and inhibiting the IP3 receptor. All of these collectively decrease the amount of calcium. Calcium is one of the stimulators of myosin light chain kinase, but hey, we don't have any calcium anymore. So myosin light chain kinase isn't active. Instead, myosin light chain phosphatase is activated by protein kinase G. If we don't phosphorylate myosin, it can't interact with actin, and we don't get any smooth muscle contraction. Instead, we get smooth muscle relaxation. That's what is going on in this diagram here. So there's a few ways that neurons can cause relaxation of vascular smooth muscle cells and pericytes when they're active. That relaxation increases blood flow, and that makes good sense. We had active neurons. They're running up a bill. The only way to pay that is with blood. Now, there's a lot of potentially toxic stuff floating around in the blood. So when we bring blood into the brain, we don't want to just dump blood on brain tissue. First of all, that'll kill neurons. Second of all, there could be some toxic stuff in our blood. So we have blood-brain barriers to prevent what can enter the brain. We want to select what's allowed in there. Things like neurotransmitters, no thank you. Those do not cross the blood-brain barrier. Hopefully we can keep all the bacteria and viruses out of the brain, so we don't get any sort of encephalitis. That never ends well. Instead, we just like to allow gas exchange, and we like to pull some glucose out. That way we can pay the bill. The blood-brain barrier will allow us to do that. Now, there are two components to the blood-brain barrier. When you hear blood-brain barrier, usually what people are talking about is the blood ECF barrier. ECF is extracellular fluid. You can also call it the interstitial fluid, the ISF, whatever. But that's what's going on over here. The blood-brain barrier is formed by tight junctions between the endothelial cells of blood vessels within the brain. So not only are our veins different, the arteries and arterioles and capillaries, these are also different. They have tight junctions to ensure that anything floating around in the blood can't just freely diffuse between those endothelial cells and enter the brain. So that's our blood ECF barrier. What's in the blood can't freely enter the extracellular fluid. We also have a barrier in that choroid plexus, the blood CSF barrier. So here we have these fenestrated or permeable blood vessels like you'd see in the periphery, so stuff can freely exit the blood. But here it encounters the ependymal cells. And the ependymal cells here in the choroid plexus have tight junctions between them, preventing free diffusion of stuff out of the blood into the CSF. So there are a couple barriers, and what they both do is keep the CSF and the extracellular fluid free of junk. Now, the structure of our barrier system is just simply tight junctions. This is what makes it a barrier. These are different from your typical adherens junctions, that's this AJ here, where there's actually a space between these two membranes. The tight junction is shown here. And notice there's not a lick of space between this aspect of the endothelial cell and this aspect. So this fuses the cell together so that what's in the blood can't freely diffuse out. It must be transported by a transporter protein. This little blow up is right here. This is just a higher resolution image. This is just showing us some of the proteins involved in forming tight junctions versus the adherens junctions. We don't need to worry about that level of detail. What we need to appreciate is that the barrier is created by tight junctions that restrict diffusion. And we find them either in the vascular endothelia, that's our typical blood-brain barrier, or in the ependymal cells, the blood CSF barrier. So if we're going to get into the brain, we have to be able to cross the blood-brain barrier. Now, some things can do that freely. For example, oxygen, right? This is a very small molecule. It's nonpolar, so it can freely pass through membranes. You can't stop oxygen from moving. The same thing is true for carbon dioxide. Still small, still nonpolar. It can freely cross. This is one half of what we need to do, allow gas exchange. We don't really need a barrier for that, right? However, there are other things that are slightly larger that we would like to transport into the brain. Things like glucose. Glucose is how we pay the bill. Transporter proteins are critical here. So the tight junctions are great because they restrict diffusion of anything that's not incredibly tiny. But if we want to move something, we need a transporter protein. This is very efficient. We can also do receptor-mediated transcytosis. This is useful for bringing insulin into the brain. We can also just engulf it. We don't necessarily need a receptor. But let's focus on our transporter proteins, because this is how we're going to transport glucose into the brain. And that's how we're going to pay the bill. Now, those tight junctions can sometimes be damaged in the case of traumatic brain injury or some neurodegenerative states like your dementias. They cause loosening up of tight junctions, and this creates further problems for the brain because toxins might be able to sneak their way through. For everything else, though, we got to have a specific protein to transport it across. So we're going to facilitate the diffusion with these transporter proteins. The most important is glucose. So we eat, we digest, we put glucose into our blood, and now we've got to get this somewhat large and very hydrophilic molecule out of the blood into the brain. And that's with our GLUTs, the glucose transporters. Sure, there are different glucose transporters in the endothelial cells. They're slightly larger than those in astrocytes. And there's a different version again for neurons. But this neuron here that ran up his bill, right? Remember, this is going to spit out carbon dioxide, potassium, and nitric oxide, all of which are going to increase blood flow, increasing the amount of glutamate that's flowing through here. So this neuron's run up a bill now. In order to get it more glucose, we have to transport it across 1, 2 membranes. Neurons don't come in contact with blood vessels. That's the job of astrocytes. 3, 4, and now it's finally floating around in the extracellular fluid to be transported into neurons to pay the bill. All right, and that's about enough of an overview of blood supply. Let's see if we can put some of this stuff together here in a few simple schematics. All right, let's start off with our circle of Willis. We should be able to recreate this. And let's start off with the input, the internal carotid arteries. All right, so these are going to come in, and let's make our blood vessels red. So a couple of internal carotid arteries, right? They'll have some branches that we don't care about. Well, you know what? I'm not even going to label it. It's the ophthalmic branch, though. We don't care about it for this class. Take that as you will. Okay, from here we're going to branch, forming the anterior cerebral arteries, which we will label in just a little bit. We also need to branch to form our posterior communicating arteries, and we'll continue along as the middle cerebral artery. This will have a branch for the deeps. That'll be my lateral striate artery. And we'll continue along as the superficial branch here. All right, good enough. Same thing over here. Let's get our posterior communicating. We'll continue along. We'll have a deep branch. You get the point. All righty. So let's label a few things. So we got our middle cerebral artery here. Same thing on both sides. We got the anterior cerebral arteries here and the posterior communicating arteries over here. All right. So let's connect the top. So our anterior cerebral arteries are going to run on up, and they're connected by the anterior communicating artery. From here, we'll have a deep branch and a superficial branch. Do a slight little bit of labeling here. Anterior communicating. And then we've got our superficial branch. This will, of course, hit more of the medial and superior aspects of the frontal and parietal lobes. And then we have our medial striate. This serves the internal capsule. And the basal ganglia. Okay, fun fact here. The medial aspect of the frontal lobe served by the anterior cerebral artery, that's going to be more legs. But the medial striate arteries serve the portions of the basal ganglia that deal with the face and arms. So if we included the whole darned anterior cerebral artery, including the deep division, we hit the face, arm, and legs. We're going to see something similar over here with the lateral striate, also serving the internal capsule and basal ganglia, but in this case, more the legs. Notice lateral, legs, both begin with L. The superficial branch, well, let's not do that. Superficial branch of the middle cerebral artery serves the lateral aspects of all, the frontal, parietal, and temporal lobes for the most part. A little bit of occipital, but now notice the lateral frontal lobes. That motor map is for the face and arms. So kind of like what we saw for the anterior cerebral artery, we can hit the face, arms, and legs if we hit both the superficial and deep branches of the middle cerebral artery. Okay, let's continue along the back. So the posterior communicating artery, here we're going to come together, but it's going to attach onto the posterior cerebral artery here. And this will also have a deep division as well. And we can do a little bit of labeling here. Posterior cerebral arteries. Deep division being the thalamo-geniculate. This serves the thalamus. The STN and optic radiations that connect the lateral geniculate nucleus to the primary visual cortex. Now, the superficial division of the posterior cerebral artery serves the more medial aspects of the parietal lobes and occipital lobes. Let's see, occipital. I'm going to take a stab at this. For some reason, that word just fell out of my head, but I think that might be right. As well as the inferior temporal lobes. All right, so that covers our 3 cerebral arteries and what they do. Now let's go on to our hindbrain circulation. All right, so we're in the basilar artery now. Our first division is the superior cerebellar artery. So let's get that on the drawing here. We've got some pontine arteries that I'll just kind of put together like this. Nah, that'll look weird. We'll continue on down. We'll get the anterior inferior cerebellar artery. Little vertebral down here. You know what? I want a little more space. I'm going to travel back in time. All right. And we're back. So let's do some short. We'll do some pontine arteries here. We'll get our ICA. All right. And we'll come on down and let's divide into some vertebral arteries here. And we'll come back together, forming that anterior spinal artery. And we need a couple of branches here. Let's get us a pica coming off. And we probably need a little division here. Oops, don't know what happened there. All right, still doing it. Cool. All right, let's label some junk. Basilar artery, superior, cerebellar artery, the anterior inferior cerebellar artery, the vertebral arteries. We got the pica over here. We got the anterior spinal artery, and then we got a pair of posterior spinal arteries, and this is just a continuation of the vertebral artery over here. All right, and then the name kind of tells us what we got here. Superior cerebellar artery. This is going to hit the pons. And that superior cerebellar peduncle. I'll just write superior cerebellum. All righty. The pontine arteries. whoops, also serve, you guessed it, the pons, no doubt there. Anterior inferior cerebellar artery also gets the pons, but the more inferior and lateral portion. And, of course, the anterior inferior cerebellum. We'll let the names work for us here. Posterior inferior cerebellum. Doesn't get the pons. It's a little bit of that medulla, though. All right, we're moving on down. And, of course, the posterior inferior cerebellum. Anterior, posterior spinal arteries. I think the name says it all. All right, there's our circle of Willis. Pretty much everything right there. If you want another representation, hey, I got you. So let's think about those three major arteries of the brain. And we'll see if I can draw the medial aspect of the brain. Kind of suck at this one. Never really practice it, as you can see. We gotta have a little thalamus in there. All right. Well, you know, if I drew that left-handed, I wouldn't feel too bad, but that was my dominant hand. All righty, let's color in some areas here. So anterior cerebral artery. Let's get that as a blue. Let's make my middle red, and I guess green makes good sense for posterior. All right, so let's start with the middle. Here we just want to kind of draw a big old area right there on the lateral aspect. Notice we're getting pretty much all of the lobes. You'll see just a little tiny bit over here. Not much, right? The more medial aspects of the brain are going to be served by the other two arteries. Anterior is going to go to the front here. All right, draw you back kind of like that. Notice we're getting corpus callosum there. That's that good old alien hand syndrome we talked about way back in MedPath. Posterior hits the back. This will hit the thalamus. And then the back. All right. This will explain some of the symptoms that we see, right? So let's get our primary motor cortex over here. You remember your motor map. Your face is right here. Upper limb here. Now we're getting more into trunk. And if we wrap around to the middle, that's where you put your feet. So more common sites of stroke are, unfortunately, the middle cerebral artery, because the internal carotid usually continues along as the middle cerebral artery. Therefore, strokes more commonly are going to have weakness in the face and the upper limb as a result. Okay, how about that posterior circulation? What's going on down there in the brainstem? All right, so let's get us a good old thalamus up here. Little midbrain action. Pons, medulla. Get a cerebellum hanging off there. Anterior lobe, posterior lobe. All right, cool. Let's get some colors in here. So who's serving us? Hell, let's just put it where it is, you know? Wouldn't that make sense? Posterior cerebral artery. It's going to serve that thalamus. It's also going to hit a little bit of that midbrain. Okay. Superior cerebellar artery. I'm feeling blue. We're going to hit a little bit of that midbrain, a little bit of the pons, and more of the superior cerebellum. Hey, we got a few more Anterior cerebellum. Well, hell, let's do posterior. Get like that, something like that. That'll do us. Okay, cool. Posterior, inferior cerebellar artery hits the posterior, inferior portions of the cerebellum, as well as a little bit of that medulla. I guess we need some labels. Midbrain, pons, medulla. Although if you don't know that at this point in the class, come to my office. Okay, we need a new color. I guess we'll make it yellow. Ooh, I like purple better. Anterior, inferior cerebellar artery is going to hit the anterior, inferior cerebellum. No surprise there. As well as some of that pons. All right, look at that. And then we've got, how about we make it black? Got the good old pontine arteries serving the pons. So the bulk of that. And shoot, we're gonna need vertebral. Maybe I will go yellow. I'd rather go orange. Look at that. So the rest of the medulla, getting those vertebral arteries. Okay. Last thing, spinal cord. All right, you should be able to do this in your sleep. Get us a spinal cord cross section. Let's get some blood vessels here. Remember, we got the unpaired anterior and the paired posterior spinal arteries. Anterior, come around, serve the front. You get the point here. So this is what will give us that anterior cord syndrome if we have a stroke at the anterior spinal artery. And then the posterior spinal arteries can give us posterior cord syndrome. We should probably have some idea of the sinuses and how they drain. Let's make them blue because this is, venous drainage. So it all starts with some kind of cortical vein. And from here, we're going to drain to the nearest sinus. Which one? Whoever you're closest to, right? And notice all these arrows go both ways because we have no valves. So it could be the superior sagittal if you're up near the top. Might be that inferior sagittal if you're not near the top. Hell, it could be the cavernous sinus. That says cavernous. Might even be that transverse sinus. All right. From here, we're going to all collect in that confluence of sinuses. Of course, we're going to have to go to the straight sinus from the inferior, which also collects from that great vein for the deep drainage there. All right, but here we'll have our confluence of sinuses. So the straight will drain there, superior sagittal, the transverse, of course. How could we forget the good old occipital sinus as well? That'll drain in there. Now the cavernous sinus gets in here via the superior petrosal sinus. Hey, look at that. We've connected everything. But I'd really like to get out of here at some point. So we need to meet up with the jugular. We can do that pretty directly from the cavernous sinus by following the inferior petrosal and then finally hitting the jugular. We got other ways of getting there, though. The sigmoid sinus drains into the jugular, and the confluence of sinuses can get there via that transverse sinus. All right, this is, what am I doing? Oh, I'm white now, okay. Superior petrosal drains in there. So you can see we got options on how we get out. Everything is all connected here. So no matter where you are, there's a way of draining the blood. And the whole point is that we will never build up pressure. We've got multiple routes to drain the blood, and the veins over here have no valves. So low resistance drainage in the veins, right? If we weren't in the brain here, our veins would have some little valves here to create unidirectional blood flow. But as nice as that is, we don't need it. The head is above the heart. We'll let gravity work for us. We don't have to work against gravity. We don't want anything to restrict blood flow. That's the whole idea there. And the last thing I guess we got to go over is how neurons regulate blood flow. So let's get us a neuron over here. It's happy. It's talking to somebody over here. And it, of course, does that by firing a bunch of action potentials. So it's an active neuron. Let's give it, that's a little sweatband. So it's working hard out there. You know what? Let's have it do some curls for the girls over here. There we go. That's an active neuron if I've ever seen one. Maybe that apical dendrite's doing a pull-up. I don't know. Let's not get too silly. All right, so when we're active, what are we spitting out? Well, one thing that we're spitting out is carbon dioxide. What does that do? Well, forms that old carbonic acid, which drops pH. That drop in pH is going to affect the vascular smooth muscle. If we're in a capillary bed, it's a pericyte, which is like an isolated smooth muscle. Anyway, let's not worry too much about that. All right, so we got our endothelial cell here. Of course, tight junctions. Hey, let's not forget that. Let's get us a smooth muscle cell. Something kind of like that. All right. You know what? I don't know if I made that big enough. I think I will have some action going on here. So I'm going to give an enormous smooth muscle cell. This is exaggerated. Vascular smooth muscle cell. I made this bigger so I can have stuff going on. All right. I think there's a method to my madness over here. So this decreases the amount of calcium that we have floating around. All right. We're also spitting out potassium. So if we increase that amount of potassium here, that opens up potassium channels. KIR, potassium inward rectifying. It's just a name. Don't worry about that. It's a potassium channel. But notice pointy arrow. So what happens? We spit out more potassium from this vascular smooth muscle cell, which leads to hyperpolarization, which also decreases the amount of calcium floating around. The last thing we also spit out here, nitric oxide. All right, that's going to go in here. It's going to activate guanylyl cyclase. That's going to take some GTP, turn it into cyclic GMP, and that activates protein kinase G. All right, what's going on here? Well, a few things. Let's stimulate those potassium channels. Let's go up here and inhibit calcium channels. Right, so what it's also doing is amplifying that decrease in intracellular calcium. And let's turn on myosin light chain phosphatase. Now, what we have to appreciate here is that contraction of smooth muscle is based on phosphorylation of myosin. So myosin can exist in two states. It can be phosphorylated, or it cannot be. If myosin is phosphorylated, it can't bind actin. Look at that. They're not bound. Here it is trying to grab out, but nope. Only when myosin is phosphorylated can it reach out and grab onto actin. And then we get contraction. But that's not what we want here. We want dilation of the blood vessel. We want the smooth muscle to relax. So if we're going to phosphorylate myosin, that needs myosin light chain kinase. And what activates it? Calcium. If we want to dephosphorylate myosin, we need myosin light chain phosphatase. What activates it? Protein kinase G. So, when we have that nitric oxide spat out, we're getting rid of the calcium. So myosin light chain kinase isn't active. We are stimulating protein kinase G, so myosin light chain phosphatase is active. Therefore, we're strongly biasing toward the right here. Thus, no phosphorylation of myosin, no contraction. Instead, myosin remains unphosphorylated and we get vasodilation, increasing blood flow to the area so we can pay the bill. Oh, sorry, this could be vascular smooth muscle cells or pericytes. The difference is, where are they? Smooth muscle is more in arteries. Pericytes are more in arterioles and capillaries. Same function, though. They're contractile cells that regulate blood flow. All right, that about does it for this. If you got any questions, send them in the questions box so I know what to talk about in class. See you later.